Hi everybody, today we are going to talk about the cell cycle and regulation. So, just to get some vocabulary so that we're all speaking the same language, you might have heard this in biology, but homologous chromosomes are two similar chromosomes with the same genes but different alleles. So when we talk about alleles, we're talking about like the big A's and the little A's. And you get one homologous chromosome from mom and you get one from dad. So if you look down here, if you look at like chromosome one of these is from your mom and one of these is from your dad. And students often confuse that with sister chromatids. And sister chromatids are identical chromosomes. And so when this chromosome right here goes through DNA synthesis, it gets a sister chromatid. And so that's the picture down here is you have one chromosome and then the other one that gets made in DNA synthesis is exactly the same. So if you have, let's say, a big R on one side, you would have a big R allele on the other side. So homologous chromosomes are similar. They have the same genes, could have different alleles, and sister chromatids are identical. Okay, so DNA is amazing. Let's talk about how big it is. So DNA in one human cell is 2 meters long. And if you think about how many cells you actually have 30 trillion cells in you. That is a lot of zeros. The DNA in a single human is 2 times the diameter of our solar system. So if you unwound all of your DNA, it would go around the solar system 2 times. That's amazing. How does it all fit in our cells? Let me tell you. So chromosomes and how they coil. So it's super important that DNA coils in as tight as it can so it can fit in all of our cells because it is the instructions for making the proteins that make us us. So there's a couple more vocabulary words. Histones are the proteins that DNA coils around. And I like to think of it as like the thread would be the DNA and the spool would be the histone. And then all of that together is what's called a nucleosome. So if you look down here, here is your histone, right? And here is your DNA that's wrapping around it, the blue. And so that whole thing is called a nucleosome. Here's another picture. So if you look at DNA, we know that it's double-stranded, and then it coils, and then those coils go around the histones, and then those actually, if you look over here, those actually coil even more to get your chromosome so that it can fit inside of your nucleus. So let's talk about different division mechanisms. So we know that eukaryotes do 2 different things. They do mitosis and they do meiosis. So in this lecture, we're going to talk about mitosis and then we'll get into meiosis. So the purpose of mitosis is for organisms to grow, to replace dead or dying cells, or to do asexual reproduction. And I always remember that mitosis happens in your toses. Meiosis, its sole purpose is to produce sex cells so organisms can do sexual reproduction. And then prokaryotic organisms like bacteria, they divide by what's called fission. And so if you look over here, I just put in a picture. So you've got a bacteria and they have, remember, they have a plasmid or circular DNA. And what happens is that DNA just doubles and then the cell splits in half. So fission means to split. So it's really simple. Okay, chromosome number. So another vocab word is somatic. So somatic cells are regular cells in your body. So you have somatic cells and you have sex cells. So somatic cells are like your skin cells or your eye cells or your hair cells or all those cells, eye cells. So somatic cells go through mitosis, and the chromosome number is what's called diploid. So di. Di means 2. So 2N is how we refer to it. And in humans, we have 46 chromosomes. So 2 of each type of chromosome, because remember you get 1 from mom and 1 from dad. So again, here's what's called a karyotype, which you can see all of your chromosomes. So you get 1 from mom and 1 from dad. Now another vocab word is germ cells, which we'll talk about more in the next chapter. Germ cells go through meiosis and produce gametes, like egg and sperm. The chromosome number of those egg and sperm sex cells is haploid, half. Because that is the egg, that's all the 23 chromosome that mom's going to give you, and then you need the 23 chromosomes from dad to make your magic 46. So karyotypes you can see are either like this or they're like this. So this is a 46, so this would be before your DNA doubles, and then this would be after your DNA has replicated. And then another thing I wanted to bring your attention to was if you look at this is an X and a Y, so that would be a male. And then if you look at this one, if this would ever go away, you would see there's just 2 X's, so that would be a female. Okay, so go ahead and pause the video and copy this chart down. So the cell cycle, and then you can add notes to it. Okay, so the cell cycle is like a life cycle of the cell. And so we often just talk about mitosis, which is down here, but really the majority of the cell cycle is what's called interphase, and that's when the cell is growing, doing its job, replicating its DNA, and then getting ready to divide. So G1 is where the cell is growing. So it's just divided, it's growing, it's doing its job. So if it's like a salivary gland cell, it's secreting amylase. If it's a pancreatic cell, it's secreting insulin, right? So it's doing whatever its job is. S is for synthesis, and this is where the chromosomes double. So when you go from 46 chromosomes and then to 92. And then in G2, the cell is doubling everything else in its cytoplasm, like the Golgi body and the ER and mitochondria, and all that other stuff is getting doubled. And then finally, the cell's going to go through mitosis. So mitosis is this blue part. And remember, mitosis is nuclear division, so the division of the nucleus. And then it goes through something called cytokinesis, which is the division of the cytoplasm. So there's 4 different parts, which we'll go into: prophase, metaphase, anaphase, and telophase. One thing that's super important in mitosis is the spindle fibers. Chromosomes can't move on their own, so they need spindle fibers to grab on to their centromere, which is the little part in the middle of the chromosome, and they pull the chromosomes to the different poles. So this is just a picture of real chromosomes in anaphase, and the spindle poles are the opposite sides of the cell. Okay, so it's super important for chromosome numbers to be maintained because you want your daughter cells to be genetically identical to the mother cell or the parent cell. If those cells weren't identical, then you wouldn't have an organism that can function as a unit. So in interphase, you have 46 chromosomes, and the chromosomes are single. Okay, and then after the cell goes through S phase or synthesis, it doubles. And so here you have sister chromatids that are identical to each other, and so you have 96 chromosomes. This right here is what the centromere is. And then mitosis happens and cytokinesis, and then you have your daughter cells that have 46 chromosomes, just like the original cell, and the chromosomes are single. And again, it's important for the chromosome number to be maintained so that those daughter cells are identical to the parent cell. So again, here's a better picture of the karyotype. You can see that it's 2 X's, and so that means it's a female. Okay, so stages of mitosis. You can kind of think of them as PMAT. Okay, so prophase, metaphase, anaphase, and telophase. And I like to do something to help me remember with my hands. So, and we'll go through this. So prophase is kind of the chromosomes are condensed. They're just kind of all over the place. Metaphase, they line up. Anaphase, they get pulled apart a little bit. And telophase, they get pulled apart even more. So that's something to remember. So when I pull up each of these new phases, I want you to pause the video and draw it. Okay? So prophase. So in prophase, you can see that the chromosomes have condensed. So what happens is in interphase, the DNA is all spaghetti-like. It's not condensed into chromosomes. It's just chromatin, which is just the DNA kind of all over the place in the nucleus. And that's because we need to be able to transcribe it and make proteins, right, for the cell to do its job. So in prophase, the cell is going to condense the DNA so that it can be divided equally to the two new cells. The chromosomes have already doubled. The DNA's already doubled because we've already gone through S in interphase. The other thing that happens is these centrioles are going to start moving to the poles, and they're starting to make their spindle fibers. And then the last thing is that the nuclear membrane starts to break up. In metaphase, those spindle fibers line up the chromosomes in the metaphase plate, and that is so that you can equally divide the chromosomes to the two sides. In anaphase, the sister chromatids are pulled apart, and they go towards their pole, and then you have equal numbers of chromosomes identical on each side. And then in telophase, again, the chromosomes decondense, so they're all spaghetti-like, and they form a new nucleus. And then at the end of telophase, the cell divides. So here's a picture I wanted to show you of what real cells look like, not just the cartoons. So here's your nucleus. Here's your cytoplasm, right? And an interphase where the cell spends most of its time. And then this one, see how you can start to see the chromosomes. So that would be prophase. Here the chromosomes are lined up. So that would be metaphase. They're starting to get pulled apart, which would be anaphase. And then you can see them forming their new nucleus, which would be telophase. Okay. So I just want you to guess which one this is. So what do you think is happening here? If you guessed anaphase, you are correct. All right, so how about this one? What do you think is happening here? You guessed prophase, you are correct. If you guessed prophase for this one, you're also correct. So this would be kind of like early on in prophase, and this would be later on in prophase. If you look at these would be an interphase. And if you notice, most of the cells of this, I think it's an onion, are in interphase because that's where the cell spends most of its time. What about this one? This, if you guess telophase, you are correct. And then these two, this would be metaphase. Okay, so the results of mitosis. We get two daughter cells that have the exact same genetic information. That is super important. So they're clones of each other. They have all the information to make a functioning organism. So after mitosis, the cytoplasm has to divide, and that's called cytokinesis. And there's two ways that happens. Plants and animals have different ways of that happening because plants, remember, have a cell wall, and animal cells don't. So in animal cell division, the contractile ring is this thing. And what happens is it just squeezes, right? It squeezes around the metaphase plate, and it forms what's called a cleavage furrow, and then the cell divides. So it's pretty simple. If you think of like a balloon, and like you're making balloon animals, and you just twist it at the middle, you're not twisting it, but you're squeezing it, and then it just divides. Now, with cell plate formation in plants, cytokinesis, it forms these spindle these vesicles, right? And these vesicles are filled with what the cell wall is made of, which is cellulose. So those vesicles get bigger and bigger, and then finally you get what's called a cell plate, which is the new wall that's forming in between the two plant cells. And finally, here you get your two new cells. Okay, so let's talk about how the cell knows how fast to divide, right? Because eventually we'll talk about problems. So there's different checkpoints in the cell cycle. So at the G1 checkpoint, so you can add this to your cell cycle drawing, the cell is going to check to make sure it's the same size, it has enough nutrients, it's got the growth factors, and then is there any damage to the DNA? Now this little thing right here, cells can get caught in what's called the G0 phase. So cells that don't divide, they get to a certain point and then they just do their job and they don't divide anymore, are going to get stuck in this G0 phase. An example of this would be like your neurons, right, in your brain. After a certain point, you don't develop any new neurons. You get disconnections between them. I don't know if you can hear Mrs. McFadden next door. She's being quite loud. Anyway, so that's one checkpoint. Then interphase, in interphase, the synthesis happens, and then there's another checkpoint here at G2, and that mainly checks to make sure that the DNA replicated correctly. And if it did, then the growth factors are going to cause it to go into mitosis. And then finally, you have one here that just checks to make sure those spindle fibers are attached to your chromosomes correctly. Okay, so the things that are going to allow the cell to continue in the right way are called cyclins, and they interact with cyclin-dependent kinases. So cyclins are proteins that are associated with specific phases of the cell cycle. So some cyclins are associated with prophase, some metaphase. The more you have of the cyclins, then the more the cell's going to divide. And if you have less of them, then the cell cycle is going to slow. And cyclin-dependent kinases, cyclin-dependent kinases are what they need the cyclins to activate them. So they're specific. So just like enzymes have a substrate, right, that's going to bind to them, kinases, ACE, so it's an enzyme, these are going to attach to a specific cyclin, and they're gonna turn into something that allows the activities of the cell cycle to continue. So here's a picture that might help put this into more understanding. So you have a cyclin. We're in interphase because we're going through G1, S, and G2. So the cyclin's going to build up, and the CDK that it is associated with, these two are going to bind together, and you're going to get this MFP, which is the mitosis promoting factor, right? And as soon as these two combine, then this protein factor for mitosis is going to stimulate mitosis to happen, and then the cyclin's going to degrade, and the CDK is going to continue on, and then new cyclin's going to build up. So it's a cycle. If you look over here, it's just the relative concentration. So as the cyclin builds up to get into mitosis, then the MFP, or the mitosis promoting factor, is also going to build up, and then they're both going to go down. So when they peak is in mitosis, and different cyclins and different CDKs are going to have different places where they peak. Okay, so let's talk about disruption. So in AP Bio, you not only need to know the cell cycle, you need to know what happens when it gets disrupted, right? And that can cause cancer, or it can cause, it's almost my favorite word in biology, apoptosis, which is cell suicide. So here is a healthy cell. So here you have a normal cell, and it's going to divide and produce 2, and then those are going to divide and produce 4. So this is what should happen. Now, if too much DNA is damaged, right, then the checkpoint will see that that's happening, and it will cause the cell to basically causes apoptosis, which is programmed cell death. And so your cells will kill themselves because their DNA is too damaged, and they can't pass that on, because then you get something called cancer. So when cancer happens, if there's too much damage to DNA in regions that actually regulate the cell cycle, then the cell cycle could continue unregulated and have uncontrollable division. So some examples of checkpoint genes and proteins are called proto-oncogenes. And so proto-oncogenes keep the cell cycle going. So I think of them as like the gas pedal. And then tumor suppressor genes are another type of gene, that code for a protein that slows the cell cycle. So if you think of your car, you need a gas and you need brakes to keep it going at a set speed. So same thing. And over here you can see the normal. Right here's normal division. Here is some genetic alteration, but what's happening is if there's too much genetic alteration, then the cell genes that cause apoptosis don't work. And so that cell is going to continue to divide. Class is over. And it's going to take over the other cells. We have a guest speaker that comes in who does cancer research, and it's super interesting because his research is to try to get those genes that cause apoptosis in cancer cells to work, to be activated, so that the cancer cells kill themselves. And that would be a treatment instead of doing something like chemotherapy, which kills good cells too. So what are tumors? So checkpoint genes mutate, right? If those mutate, then cell division is, control over cell division is lost. So those proto-oncogenes, if those are mutated, they turn into oncogenes, which is like the gas pedal getting stuck. So if your gas pedal gets stuck, no brakes, and you have brakes, you're going to keep going forward. If your tumor suppressor genes get mutated, like if your brakes don't work, then you're going to keep going forward. So both of those, if they both mutate, you get cancer. So cells dividing out of control creates a mass, which is called a tumor. And some words you need to know about this are benign. So like a mole, if you have a mole, it's a benign tumor. They're not spreading. And malignant, which is cancerous. And another word you might need to know, that you should know, is metastasis, or if the tumor has metastasized, that's what the lingo is. Now, if you go to a cancer doctor, they're called an oncologist, and that's why these genes are called oncogenes. So here is just a picture of a cancerous cell, and you can see that it's spreading, so it's metastasizing, so this would be a malignant cell. And then here is a picture of a skin cancer area, and you can see that it has, the edges are kind of not smooth, and also it is red. So when you have, like I have a lot of moles, and so what you want to look for is you want to make sure that your moles are not getting bigger and changing. So if they change shape, if they change size or change color, that's when you need to go into a dermatologist. So hopefully you learned something about cancer, not cancer, well, cancer too, but hopefully you learned something about the cell cycle, how it's regulated. Make sure you have all the drawings of the different phases and what's happening in each phase, and also the drawing of the cell cycle. So have a great day.